Namaste to all. Welcome to the next session on molecular biology and our DNA technology. In this session, we are going to see about the next topic, that is organization of mitochondrial and chloroplast genome. In our previous session, we have seen about prokaryotic genome and eukaryotic nuclear genome. So we have seen that in eukaryotes, in plant as well as in animals, we have mitochondria and chloroplast. Mitochondria and chloroplast, if you see in general, they have their own machinery, that is, they have their own matrix and their DNA material inside them. They have been developed during the evolutionary process and being residing inside our cell from millions of years from the evolutionary period. Let's see about how the mitochondrial and chloroplast genome is being organized. So if you take mitochondria, they consider they are the critical role in generation of energy in eukaryotic cell. They are mostly involved in the breakdown of carbohydrate and fatty acids by oxidative phosphorylation. They contain their own genome with DNA. Organization of mitochondria, if you see in this, you can see they have the outer membrane, and inner membrane is called as lamellae, which is inner boundary membrane and crystal membrane. Then in between the outer membrane and inner membrane, we have the inner membranal space, which is called the intracrystal space or peripheral space. They have two types in this. So then they have the ribosomes. You can see here the ribosomes are there. Then granules are there. Then inside the slimy materials, which is having enzymes and other systems, that is matrix, they have their own circular DNA. And the dots, what you see here, they are the ATP synthase particles, which is helpful during the ETC, that is electron transport chain. And these are the crystase, F1 particles, which helps during the electron transport chain. So if you see the genetic system of mitochondria, they contain their own genetic system. They are separate and distinct from nuclear genome of cell. They have been thought to have been evolved from bacteria millions of years ago, evolved by endosymbiosis method. That is, prokaryotes are engulfed and living inside the cell during the course of evolution. This theory was proposed by Lin M in 1960. The genomes of living organisms that are similar to the mitochondrial genome are those of the free living proteobacteria, which codes for 6,700 to 8,300 proteins. Other organisms which can live inside eukaryotic cells are Rickettsia prowazekii, encodes for 830 protein coding gene, and transcribes and translate on their own. So if you see the evolutionary process of the development of mitochondria and chloroplast, you can see here when that is the ancestral prokaryotes, they have their own plasma membrane, DNA, and cytoplasm. This has been evolved and they have been engulfed by infolding of the plasma membrane. Due to this infolding in the evolutionary process, they became as with nucleus with nuclear envelope and endoplasmic reticulum. The cell with the nucleus and the endoplasmic endomembrane system has been formed. Later, by engulfing of aerobic heterotrophic prokaryotes, the mitochondria has been developed. In the course, these prokaryotes, are further engulfed by another photosynthetic prokaryotes, and these in the course have formed as the protoplast or plastids. So the division, if you see in this, after the heterotrophic prokaryotes, they became as animal cells or in the course with the mitochondria alone and became as heterotrophic eukaryotes. And when the engulfing of photosynthetic prokaryotes have taken place, they have become as the photosynthetic eukaryotes or ancestral photosynthetic eukaryotes, which is having the chloroplast and plastids. So let's see about the mitochondrial genome or DNA. They are circular DNA and has multiple copies. They work same as the prokaryotic genome. Human mitochondrial DNA is 16.6 kb. They are larger. Mitochondrial genomes are found in yeast approximately 80 kb and plants have more than 200 kb. Larger mitochondrial DNA has non-coding genes only, significantly contain no genetic information. So if you see the varieties, biological name and mitochondrial size and how many encodes of protein takes place in this, if you see plant, that is Arabidopsis thaliana, the mitochondrial DNA is 367 kb and encodes for 31 proteins. Protozoans, that is Racinomonas americana, that is mitochondrial DNA, is 69,034 base pairs and contains 67 genes. Nematodes, that is Plasmodium falciparum, mitochondrial DNA is 5967 base pairs, which codes for 3 proteins. And free living proteobacteria, the mitochondrial DNA is 7 to 10 that is MBP, that is base pairs. So the human mitochondrial DNA, if you see, encodes for a small component of protein. It also encodes for ribosomal RNA and most of the tRNA required inside mitochondria. It encodes for 13 proteins. Mainly, it involves in the electron transport chain and oxidative phosphorylation. Also, human mitochondria encodes for 125 rRNA and 165 rRNA and 22 tRNA. Translation of proteins of organelle genome. The 2 rRNAs are the only rRNA component of animal and yeast mitochondrial ribosomes, in contrast to the 3 rRNAs of bacterial ribosomes, which are 23S, 16S, and 5S. If you take the plant mitochondrial DNA, they have the third rRNA, that is 5S. And plant and protozoan mitochondria, if we compare, they differ in importing and utilizing tRNA, which encoded by nuclear and mitochondrial genome. Use of slightly different genetic code, which is distinct from the universal genetic code used by both the prokaryotes and eukaryotic cells. So mutation in mitochondrial DNA. If you see the mutation in mitochondrial DNA, we can understand that the adult or the elderly subjects, if the skin in that the mutation has taken place, that is iPSC, pluripotent stem cells 1 and 2, that is the somatic mitochondrial DNA mutation, then it is mitochondria is affected and there will be no functions for the energy generation during the ETC. In the same manner, the blood which is there, in that if the iPSCs, that is the I pluripotent stem cells 3, in that the division can happen. In some cases, they can become normal. And oocytes, if something happens or mutation takes place, the normal mitochondrial DNA, there will be sometimes it will be normal. So all the mitochondria of fertilized egg contributed by oocyte. So germline mutation, so transfer to the next generation by mother's oocyte alone. Mutation in one tRNA in mitochondria leads to metabolic syndrome, that is further leading to obesity and diabetes. Optic neuropathy, that is leading to blindness. Mutation in mitochondrial protein required for electron transport chain. Mitochondrial protein half numbers are still unidentified, that is heme biosynthesis in bone marrow. Let's see about the chloroplast DNA. Evolved by endosymbiosis and larger than mitochondria. Function, that is generation of ATP in plants, conversion of CO2 to O2, chloroplast synthesis, that is amino acids, fatty acids, and lipid components of their own membranes, reduction of nitrate to ammonia, essential step in the incorporation of nitrogen to organic compounds. Let's see about the chloroplast. So if you see the chloroplast structure, they have the outer they have the outer membrane, inner membranous space, an inner membrane which is called as chloroplast envelope. Inside them, they have the thylakoids, which are the lumen thylakoid space with low pH, and thylakoid membrane is present over them. They have been joined and they are being stacked one above the other to form the granum. Between the one granum and another granum, the bridge is actually called as lamella. Inside them, the slimy or the moving material which is present inside like a matrix, this is called as stroma, which is of high pH. They have their own DNA ring which is present inside the stroma. They have ribosomes, lipid granules, starch granules which are present. So if you see the chloroplast genome, they own genetic system as they have their own genetic system as in mitochondria. Evolution from the photosynthetic bacteria by endosymbiosis. The 6 to 9 MB genomes of present day free living photosynthetic cyanobacteria code for between 5,400 to 7,200 proteins. They have circular DNA, which is it is present in the stroma. They have multiple copies which are present and larger and more complex than those of the mitochondria, ranging from 120 to 160 kb and containing approximately 150 genes. It encodes both RNA genes and for functional proteins involved in photosynthesis. Chloroplast genome. So if you see the chloroplast genome as compared to the mitochondrial genome, I'm taking the example of tobacco, that is Nicotina tabacum. The chloroplast DNA which is present is 1,55,939 base pairs. 4 rRNAs are present, that is 23S, 16S, 5S, and 4.5S. They give for 30 tRNA species. Chloroplast tRNA, it is all translate all the mRNA codons, 20 ribosomal proteins, that is 1/3 of the chloroplast protein, some subunits of the RNA polymerases, and also some factors involved in transcription are encoded. One of the subunits of ribulose bisphosphate carboxylase, that is Rubisco, is encoded by chloroplast DNA. During Calvin cycle, enzyme catalyzes addition of CO2 to ribulose 1,5-bisphosphate. So I hope that in this session you have got an idea about how the evolutionary process for the development of mitochondrial genome and chloroplast genome has taken place, and what are the different features of mitochondrial DNA and chloroplast DNA. We will see in the next session with more detail about prokaryotic replication and eukaryotic replication. See you in the next session. Thank you. Bye.